Lesson 7 is about DNA and heredity. DNA is the substance that we all have in each of our cells within the nucleus, as we discussed earlier, and that is really the blueprint for the entire cell. Gives the ability of all the other cells in the body to carry out their functions. Heart cells to pump blood, eye cells to be able to see, etc., etc. And DNA stands for deoxyribonucleic acid. It's a really complicated word, but if you break it down, it's not so complicated. And really, it's a series of what are called nucleotides. A nucleotide is simply a combination of a base, a phosphate, and a sugar. Most sugars, like the glucose we learned about earlier in the course, have 6 carbons, whereas the sugar in DNA has 5 carbons. So, and a nucleotide, which is really the building block for DNA, has a 5-carbon sugar, a phosphate, and a base, which is a molecule that has nitrogen in it, hence its name, the nitrogen base or nitrogenous base. Here's an example of a nucleotide. Adenosine, we actually saw adenosine before when we were talking about ATPs and ADPs, but it's also an example of a base that could apply in a DNA molecule. You've got a sugar over here. Notice the 5 carbons, 1, 2, 3, 4, 5. And you've got a phosphate group over here, which has a phosphorus in the middle, surrounded by a bunch of oxygens and hydrogens. So that's what DNA is. DNA is a nucleic acid, which means a bunch of nucleotides. Notice that word there. And the reason why it's called deoxyribo is because it is a ribose sugar that's missing an oxygen. We'll see a little bit later on there's something similar called RNA, which is ribonucleic acid, which is not missing an oxygen. Anyway, so it's called deoxyribonucleic acid, and a nucleotide, as we saw, has a phosphate group, a sugar called deoxyribose because it has 1 less oxygen than a comparable sugar called a ribose, and it also has a nitrogenous base. And there are 4 different possibilities: A, G, T, or C. Try to remember the letters. You don't necessarily remember the names. The names are adenine, guanine, thymine, and cytosine. And interestingly enough, it's really just in the order of those bases that makes a determination in terms of what's going to happen to the cell and with the cell. The DNA is shaped in sometimes what is known as a double helix. We're going to see a little bit on the next slide. A double helix is kind of like a spiral staircase with stairs being the bases. Here's some examples of some representations of the double helix. These are just different types of diagrams, different examples. The backbone over here, this line and in this diagram it would be this line and this line, consist of the phosphate group and the sugars, and these little bars going out to meet each other are the bases. In other words, there's one on this side and one on this side, and this base is moving to meet with that base. That's why it's called a double helix, because there are 2 strands of DNA that are kind of wound around, and they're facing each other, and each nucleotide meets its opposite nucleotide, the one on the other helix, with a base that extends out to the other one. Okay, so let's take a look at the slide. Over here, the DNA ladder is like a spiral staircase with 2 strands wound around each other, and the bases are held together with hydrogen bonds. They can be broken by some sort of a drug or medication, but again, I don't even want to really go into that right now. But the point is, one important thing is that, notice, as we discussed before, this is the backbone of it, which contains the phosphates and the sugars, and these things over here are the bases. Now, the one thing that is important is that A only bonds with T, and C only bonds with G. So if you can remember those 4 letters, A, T, C, and G, which is adenine, thymine, cytosine, and guanine, the C only goes with the G, and the T only goes with the A. So for example, in this diagram over here, the green thing being adenine, the yellow thing being cytosine, purple guanine, and I guess maroon thymine. So you can see here is the A and the T, this is the C and the G, AT, GC, GC, AT, etc. You can see the A's always go opposite the T's, and the C's always go opposite the G's. That's the way the double helix is structured. Here's another representation of it. Probably a better diagram over here. On the top just represents the fact that you have the A's opposite all the T's and the C's opposite all the G's. And on the bottom you have the same thing but in the form of a double helix, which is the way the DNA really operates. Kind of two strands winding around each other. Helix is really just another word for a spiral. Now, the thing that DNA is important for is that the DNA is the thing that codes your genes. We're going to see a little bit later on. Each 3 bases of DNA are able to encode one amino acid. And how that combination, we've got millions and millions and millions of bases of DNA in each of our cell, and that combination, the one that codes the appropriate amino acids, makes the difference between somebody who has brown eyes and somebody who has blue eyes, or between a human being and a fish and a goldfish, is really just the base order that's in our DNA. Now we happen to have different amounts of DNA also than goldfish do, but because the DNA is what programs the amino acids to do what they do. So genes are made of DNA, and DNA encodes genetic information. The way the DNA actually works is that it produces 2 double helixes, or double helices, with the same genetic information on both, and that's how the cell replicates. Remember when we discussed mitosis, we discussed how the chromosomes replicate. The chromosomes split apart into 2 identical chromosomes, and they each eventually go and form part of the child, part of the offspring. RNA is the partner of DNA. The DNA essentially just stays in the nucleus, doesn't go anywhere. It's kind of like the blueprints that you're not allowed to take out of the library. You can go to the library and look at the blueprints, but you can't take it out of the library. If you want to get a copy of the blueprints, you can photocopy the blueprints in the library and then take it out. Well, that would be the RNA, because the RNA, in essence, photocopies the DNA and moves it around to different parts of the cell, to the ribosomes where the amino acids are being made, and to whatever other parts are to the mitochondria, whatever other parts are necessary to be programmed by the base sequence that apply in the DNA. So the DNA itself cannot actually synthesize the proteins like in the ribosomes. Instead, it uses the RNA to get its message out. RNA, as you can imagine, is very similar to DNA. It is also made of nucleotides. It is also made of sugar, phosphate, and base groups. There are a couple of subtle distinctions between DNA and RNA. You might want to remember these. These could be important. It has a backbone of ribose instead of deoxyribose. Remember, the nucleotide for DNA had a sugar, base, and a phosphate, and the sugar was called deoxyribose, whereas the RNA also has a sugar, phosphate, and a base, but the sugar is ribose. As you might be able to imagine, the only distinction between ribose and deoxyribose is that the deoxyribose has 1 less oxygen, hence the deoxy. The other distinction is that instead of the T, the RNA has a U, uracil. So instead of the T going with the A, it's the U going with the A. And the C and the G are the same. That's just, again, one subtle distinction between DNA and RNA. Now, the way RNA works is essentially what you have. I'm going to skip to the slide before and skip the bottom for the time being. You have 2 types of RNA. You've got the mRNA, or the messenger RNA, and they basically photocopy or get attracted to the bases of the DNA in the nucleus. Let me find a diagram like that. Here we go. This is a good one. Let me show it to you over here. Okay, this is a classic example of transcription. You've got this stuff, this dark blue material over here, and that's the DNA. Now, you notice how in the DNA you've got lots of bases, A, C, T, G, A, T, C, etc. And they all have the opposite bases over here. T opposite the A, C opposite the G, A, T, C, C, D, etc., etc. So that was the original DNA double helix, and what happens with transcription is the DNA kind of splits apart. You can see these two kind of came apart, and the bases of the DNA attract RNA bases that are their mirror image. The T's attract A's, the A's attract U's. Remember, there are no T's in RNA. This green strand is an RNA. The C's attract G's, and G's attract C's, etc. So now if you notice, once this happens, you've got an mRNA strand, a messenger RNA strand, and that is the exact mirror image of the DNA, because the bases are the exact opposite. Then the mRNA goes out into the ribosomes, or goes out into wherever they need to be, and they attract another set of RNA called a tRNA, called the transfer RNA. And if you notice, that's really kind of like a double image. The DNA attracts the mRNA that's the opposite of it. Then the mRNA attracts the tRNA that's the opposite of it. And so the tRNA are essentially going to be the same thing as the DNA, which of course, with the exception of the U's being instead of the T's. So the mRNA carries the information from the DNA to the cytoplasm, in other words, to the outside of the cell. And this is kind of a copy of the DNA. And the transfer RNA then gets encoded by the mRNA and essentially is the same coding as the DNA. And then what happens is that each three bases, each pair of three, well, really a link of three bases, is enough to encode one amino acid. An amino acid is the building block of proteins in general, and a three-base sequence is called a codon. And the reason why it's called a codon is because it encodes one amino acid. And a group of amino acids, when you get a whole bunch of amino acids together, they make a polypeptide, which, as we learned earlier in the course, is really the same thing as a long protein chain. And so then the mRNA is now capable of, the tRNA is now capable of, wherever it is, and the ribosomes are out in the nether reaches of the cell, wherever they happen to be, of encoding these codons. And the codons then determine what kind of amino acids are formed, and what kind of amino acids are formed determine what they're actually going to go out and do. Where problems arise is sometimes referred to as a mutation. A mutation is what happens when a DNA sequence gets messed up a little bit. Base sequences get altered, some of the bases get cut out, some of the bases are doubled, some of the bases are halved, whatever the case may be. Mutations is when there's some sort of a problem. Now, these happen all the time, and they're typically fairly harmless. If it's bad enough for the cell to have a problem, the cell can just be killed off and replaced. Obviously, a lot of mutations, or really bad or really unlucky mutations, could cause serious problems for a person, but mutations in general are fairly routine. So if a base pair is mismatched, then the mismatch can simply be cut out, and it can just be reformed in the correct way. However, sometimes the enzyme replaces the nucleotide parental nucleotide instead of the mismatched one. And so what could happen is, because of a mutation, it turns out there's some sort of a mismatch in the DNA or in the RNA, whatever is encoding the amino acids. And because of that, what that might happen is it just could change the function of the cell. And the cell, it doesn't have the right amino acids and either can't function or functions in a different way. There are different types of mutations. Insertion is when an extra base is inserted. Deleted is when a base is deleted. Inversion is when bases are backwards. And translocation is when they're changed places. And a base deletion near the start of a coding sequence, let's say near the start of the sequence that codes what's going to happen, what a person's eye is supposed to do, that's obviously the worst kind of mutation. So this is just some examples of different types of mutations. Point mutation is where you have, let's say, the wrong type of base gets inserted over here. Insertion is you have an extra base, this T over here in the middle. Deletion is when this A base got deleted. Point mutation, you can see, these are pretty self-explanatory. And these could cause diseases and could cause serious problems, especially when you're talking about individual like a zygote, a brand new baby cell that doesn't have enough cells in the rest of the body to overcome it. And mutations could cause obviously very dangerous things like genetic diseases and even cancer. And these bases are the format for genetic expression. The appearance of a phenotype, like brown eyes or black hair or whatever it is attributed to a particular gene. In different tissues, different genes are expressed. Even though your DNA in each cell has what is necessary to program the entire body, in your eyes, only the cells that affect eyes are expressed. In your heart, only the DNA that affects the heart is expressed, etc. Because your eyes don't grow hair, because different genes are expressed in different tissues. Your hair-growing genes are not expressed in your eyes, thankfully. Okay, so let's take a look at a couple of multiple choice questions for this chapter. A sequence of bases in the DNA section is AA, TC, GG, AT. What is the corresponding sequence of mRNA? Well, remember, you always have U's instead. It's RNA, so you always have U's instead of T's. So the A would correspond with a U. So just looking at the first three, AAT would have to be UUA. And I can see there's only one choice that starts with the UUA. Let's just make sure the rest are correct. CGG would be GCC, which it is. And then AT would be UA, which it is. So C would be the corresponding example of base sequences to this one. Next one, which of the following has the fewest nucleotides? Okay, well, mRNA strand having 100 nucleotides, okay. mRNA having 75 codons. Now remember, a codon has 3 bases, and each nucleotide has 1 base. So something that has 75 codons would actually have 225 nucleotides, 225 bases. Protein having 50 amino acids. Now amino acids are programmed by 1 codon, so that would actually have 150 bases, 150 nucleotides, so that's more. So far the least is A. A protein with 10 polypeptides having each 40 amino acids, that's 400 amino acids altogether, that's obviously a lot. And an mRNA having 100 bases. I don't know what it looks like. I'm just going to cut choice E because choice E looks like the same as choice A. So anyway, the correct answer, if we cut choice E, is just plain choice A.